Hello my dear students, I hope all of you are doing very well. So, we are learning immunity, immunology, how vaccines are working and all those things are associated with your immune system. So, today I am going to tell you about the structure and function of MHC, how the MHC is working and what is the role of antigen presenting cell and how basically T cells are working, everything I am going to tell you on today. So, the things that I am going to discuss with you, I will tell you the antigen presenting cell, then MHC, that is measure histocompatibility complex, then structure, then how it is actually working, various genes associated with MHC and what are the similarities between two different classes of MHC, that is class 1 and class 2, what are the differences and how cytokines are actually taking a big role in your immune system. So, let me tell you before entering to the main concept, I want to remind you that in my first lecture I have mentioned that we are actually surrounded by all those microbes. So, if those microbes may enter in your body, you have the first line defense, it is called physical or physiological barrier, then you have second line defense called innate immunity and you have third line defense, it is called adaptive immunity. So, at first this first line defense will come to the picture. If they are not able to stop the viruses or bacteria or the pathogens, then the second line defense will come to the picture. If they fail, then the third line defense will take to the matter and it will save your body from the viruses. Now, in previous lectures I already mentioned that in adaptive immunity, that is third line defense, there are two parts or there are two different ways. One is the T cells and another is the B cells. I hope you can remember in my last lecture I have mentioned you how B cells are working. B cells are producing various antibodies and lots of immunoglobulins and they are saving our body from those pathogens. Now, today I am going to focus here, that is T cells mediated immunity, how T cells are actually come to the picture. You see, when microbes will come here, when all those cells like neutrophil, basophil, eosinophil, macrophage, mast cell, they do not able to control by themselves, then they have to report about the pathogen to the T cells. How they will report? That is the matter of understanding. So, these are your microbes or pathogen. When, for example, this is your cell, this is your cell. So, if any of the pathogen may come and it may struck to your cells. So, what will happen? For example, macrophage, it will engulf that thing, it will take those pathogen within its body like this, and after that there are lots of enzymes and lots of chemicals are present in that particular macrophage. So, it will chop it out, it will chop it in various pieces and then this macrophage it will present those pieces as an antigen to whom? To T cells. How? Let me show you. You can see one by one whatever the chopped pieces was there, it is coming here, it is actually showing to the T cells, so that they come to the picture and they take the action. So, first T helper cell, they are actually showing to the T helper cell and T helper cell actually making one bond or complex. So, at that time it will also release various kinds of chemicals like interleukin, and then when interleukin will be released, interleukin will again it will inform the surrounding other cells like cytotoxic T cells and B cells. Why? Because B cells will generate lots of antibodies, B cells will generate lots of antibodies and then the action will be happen. Whatever I have shown you, let me play with a video in this, just a second. Okay, it is okay. It will be not properly playable video. Okay. Anyway, so I hope it is very much clear to you all that how our the macrophages are actually presenting the antigens to our T cells. So, this complex is very important, because from this complex our MHC, what I have discussed at the beginning of my lecture, MHC and our antigen presenting cell, the concept is coming. So, this cell will be actually known as the antigen presenting cell. So, in definition, antigen presenting cell are the cells or a group of immune cells that mediate the cellular response by processing and presenting antigens for recognition by certain lymphocytes such as T cells. Classical antigen presenting cells are like dendritic cell, macrophage or other kinds of cells. I have mentioned about the specifically the macrophage. You have seen in the picture, I have mentioned that how macrophages are actually working. Now, let me tell you this area, let me remove the inks. Okay. This area we are actually calling as a MHC, major histocompatibility complex. Now, let me tell you more. Now, you have seen that macrophages are actually working as a antigen presenting cell. So, we can categorize the APC, antigen presenting cell, to two ways: professional APCs and atypical that is APCs, like dendritic cell and macrophage, or B cell, we can call them as a professional antigen presenting cell, or others like mast cell, basophils, eosinophils and interleukin that is a C3 type, we can call them as a atypical APC. Let me tell you more. So, this complex, basically this complex, so majohistochondrial complex, it is a group of genes that code for the proteins found in the surface of the cells that represent the antigens to T cells, clear. So, without representing, you can imagine if your the macrophages does not represent the antigens to your T cells, then how your immune system will be activated and how your immune system will proceed, that is adaptive immunity. That is why this complex is very much important, and it helps to the recognition and discriminate between the self and also non-self. One of the major that is a property of our immune system is that it can differentiate between our own immune cell and a foreign substance. Let me also tell you about the antigen presenting cell. Another function of the major histocompatibility complex is the tissue that is a allorecognition, which is an important factor in prevention of the successful organ transplantation. If any organ transplantation may happen in future, you will be observing that from heart, from liver, every organ we can transplant. Why? Because of the advancement of 3D printing organ technology. So, that is why antigen presenting cell will again come to the picture. So, the human being is the complex also called as a human leukocyte antigen system, HLA system, we also call it this. Now, come to the types. So, you can categorize into two ways. One is the MHC class 1 and MHC class 2. Now, why there are two different classification? So, by this picture I will tell you. For example, you have cytotoxic T cell, you have cytotoxic T cell, cytotoxic, cyto means cell, toxic means it will be toxic. So, cytotoxic T cell it will be going to bind to the infected cell. For example, SARS-CoV-2 entered in your body, it is infected you, it has infected your one cell. So, cytotoxic T cell will destroy that infected cell, but helper T cell will be going to the antigen presenting cell, they are not actually destroying the entire cell. So, here is the differences. So, when your infected cell, it will represent through MHC, it will represent through MHC, infected cell will mix with the or bind or make a complex with the cytotoxic T cell. So, in this area what those kinds of MHC will form, we will call it as a class 1, and in this case if any that is only microbe is present, cell we are not going to destroy that cell. In that case, when the complex will happening with helper T cell, in that time this complex we will call it as a that is class 2. Why class 1 and class 2? Here is the answer. You can see here is only one and here is two different that is bond, that is why. So, this is the entire picture. If this is your transmembrane segment and this is the peptide binding cleft, and you can see how they have actually arranged. So, because of only presence of two things, two different bonds, we call it as a MHC class 2. So, differences. So, MHC class 1 molecules are found almost all the nucleated cells of the body, and MHC class 2 molecules are found almost antigen presenting cells like macrophages, B cells. Okay. So, that is the differences between these two. And if I tell you more, for example, here is a typical picture how the interactivity is happening. For example, this is a cytosolic protein. First antigen uptake was happen, then antigen processing through the that is a proteosomes, and some peptide was actually formed, then this peptide actually came to here, the peptide MHC association, and then it is showing here. So, at that area you can see this is our T cell receptor, this is CD8, and here expression of the cell surface. On the other hand, if any extracellular protein is actually coming here, uptake, and then it was processed, chopped, and then it is going, and here you can see again one bond was formed like this, but here is a double bond, that is the differences. And if I tell you more, the genes from where it is usually coming, this is the entire picture. I will not discuss too much, but I would like to mention that another class is also present, that MHC class 3. Many of the books it was not actually properly mentioned, they try to mention only MHC class 1 and class 2, but it is also important that you should know MHC class 3, though it is not, we are not so much giving focus on MHC 3, but we should know about this. This is the chromosomal that is a diagram where from where actually it is coming. Now, in human, the MHC proteins are included by the cluster of the genes located in the chromosome 6. The picture whatever I am showing you, it is from the chromosome 6. So, whatever I was mentioning you that there are 3, I have discussed with 2, but actually it is 3. But the MHC class 3 we are not giving so much importance to that. There are lots of new research has to be done on this area, and among those MHCs, the 2 types of are actually most popular, class 1 and class 2, whatever I already mentioned with you. Now, let me tell you what are the similarities. So, both are actually synthesized in the rough endoplasmic reticulum, ER reticulum. Okay, both are found in the surface of the antigen presenting surface. Both are enclosed by genes in HLA locus sorry, and in both class 1 and class 2 expression of the gene is codominant. If I tell you more some differences, so peptide binding domain, for this case it is actually alpha 1 and alpha 2, but here it is the alpha 1 and beta 2. Amino acids, how many amino acids will be present? For this case 8 to 10 amino acids will be present, but in this case it is a bigger, that is why 12 to 18 amino acids. You can see in this picture, you can see in this picture here are another bond is happening here, and you can see this place is empty. So, you have to count and you have to wait for that place also. So, that is why more and more amino acids will be present there, that is why from 10 it is actually rising to the 13. Distribution, all nucleated cell, and MHC class 2 it is present only to the macrophage, B cells and dendritic cells, already I have mentioned that thing. Location, it is present antigen to the cytotoxic T cells lymphocytes, and it is present only to the helper T cells lymphocytes. Okay. And binding site for T cell receptor CD8 bind to the A3 region, and here it is binding to the B2 region. So, these are some differences. If I tell you more, then it is actually binding with binds endogenous antigen synthesized in a cell, it is bound to the exogenous antigens, exogenous means outer, and it acts just a second, acts, how it is actually acting. So, presence of foreign or our abdomen that is antigen target cells destruction, and it is actually working for the presence of the foreign antigens. For example, some virus actually entering, some bacteria is actually invading, it is destroying your cell. In that case, it will come to the picture. Chains, it has that is a E variant chain, and it has no E variant chain, and it has E variant chain. We have seen there are 2 chains. And so, after discussing this part, after understanding our all the antigen presenting cell and how they are actually presenting some microbes to our T cells, so the T cell can take some action. Now, the biggest question is that communication, how they are actually communicating with each other. You have seen that our B cells, and you have seen our that is T cells, T cells. So, B cells are producing your antibody, and you have seen T cells are directly going and binding like through cytotoxic T cell. I am writing as a cytotoxic T cell. So, that is the major differences. Now, the question is how both of them are actually communicating with each other. So, the answer is cytokines. Yes, there are other chemicals also, but majorly we are actually focusing on cytokines. What is cytokines? Small soluble molecules. Okay. Cytokines was in news in recent times, because after the that is outbreak of COVID-19, lots of patients actually have died because of the cytokine storm. I will tell you more how it is actually happening. So, what is cytokine and how it is actually working, and let me tell you something. So, for example, you have that is a simulated cell, it is producing some cytokine. So, cytokine actually secreted here, and the specific cytokine receptor on the target cell bound and it is activated. So, this is one cell and this is another cell. It is actually communicating, it is sending some information to this cell through the cytokines, it is by secretion of the cytokines, and then it will be, this cell will be getting activated. So, in definition or in detail, cytokines are produced and secreted by the wide variety of cells. They act as an intercellular mediator, like hormone like substances. That means they are signaling molecule, hormone like substances. If I tell you more, cytokines are also soluble molecule, and some of them are membrane bound molecules. Okay. So, let me I have lots of things to inform you about cytokines. So, chemically they are actually can be protein or glycoprotein, and their molecular mass should be less than 30 Dalton, 30 kilodalton. Okay. It means these will be very small, very small. Cytokines have very short half life, and mostly the cytokine act over the short distance, like autocrine or paracrine. Now, what is autocrine and paracrine? Autocrine means the cytokine acts on the same cell, and the paracrine means the cytokine acts on the nearby cells. Now, let me tell you an interesting story. So, drugs, our drugs, according to Lipinski rule, drugs should be for a drug, its molecular should be less than 500 Dalton, and here you are observing the size of a cytokine, how much smaller it is. That is why for a disease, if only cytokine is actually responsible, it is very difficult to target by using an interact molecule. And you have also seen that it is actually working, or it is actually targeting in the localized area, and these are effective at low concentration. This is because of the specific high affinity of cytokine receptor. So, some receptor you can see this area, it will bind only particular that receptor, it will not bind anywhere. Okay. So, its size should be small, and it will not able to go too much that is far away from the site of action, and it will directly work and it will bind with the receptor. So, I was telling you the cytokine storm in COVID-19. So, why in COVID-19 cytokine storm is actually happening? All of you might have seen in news reports, and as a student of pharmacy, you also might have seen that patients are actually suffering from these various symptoms like fever, impaired hematopoietic function, and sometimes dismented intravascular that is a coagulation, or decrease in the serum protein, and other kinds of symptoms you also might have seen. How these symptoms or how these problems are actually happening? The answer is actually associated with the cytokine and its storm. For example, coronavirus, this is what? This is our lung, I am writing as a L, this is our lung. So, when a coronavirus that is SARS-CoV-2 entering in your body, what it will do? It will first go at the lung cell, like AKtU receptor is present, it will bind with the AKtU receptor, and then when it will bind, it is a foreign substance. This SARS-CoV-2, it is a foreign substance. So, your entire immune system will be get activated. So, macrophage, dendritic cell, T cell, fibroblast and endothelium, all the things will be activated. Let me erase, all the things will be get activated, and I have mentioned you that to communicate with other cells, they will actually release some cytokines, they will release some chemicals. So, macrophage, it will release these things, other cell it will release these things, almost if you may just see this graph. So, all these various kinds of cytokines will be get released. Due to that, all these various cytokines, it will be actually responsible, it actually releasing to inform the other cells that you may get activate, because our body is get that is invaded by some foreign substance. But there are some side effect you cannot say, or some other kinds of symptoms, clinical features you can say. So, these things we are actually getting. So, basically they are doing their work, cytokine is doing their work perfectly, they are informing to the other cells, other human cells, that you may come to the picture, you may go to the particular that is a war zone where SARS-CoV-2 actually destroying our body cells, and it is replicating itself, and it is making its number so very large. So, that is why it is very much important to know how the cytokine storm is actually creating a big role. So, in future if you see this kinds of that is a feature or this kinds of problem, so you have to understand that somehow it is associated with our immune system, immune cells. So, here is another picture I have brought for you people. So, you can see, you can compare this to alveolus. All of you are aware, through alveoli our oxygen is actually get absorbed and carbon dioxide get released. So, this is a healthy and this is a injured. You can see there are lots of that is a liquid are actually present here, and your all the immune cells are actually get activated, they are working here to kill the virus. But for your body, if the virus is new, it is not possible to your immune system to develop antibodies. You need the antibodies to kill the SARS-CoV-2 like virus. That is why your alveoli will get affected, and after some time it will be not working properly. So, what are the various types or what are the various cytokines? First is interleukin, interferon and chemokines. So, let me tell you more interleukin. So, it is a subset of a large group of cellular messenger molecules called cytokines, about which I was actually telling you, which are modular of the cellular behavior. Like other cytokines, interleukins are not stored within the cell, but are instead secreted rapidly and briefly in response to the stimulus such as infectious agents. Interleukins actually, there are lots of types of interleukin. People are doing their not only PhD, they are doing their postdoc to discover a new interleukin. So, I will not confuse you or I will not tell you details of interleukin, otherwise you will feel this lecture is very difficult. So, for today as a student of B Pharma, you have to just understand that it is a part of our cytokine. There are lots of types, lots of ways of interleukin. So, these are various types of interleukin and how they are actually functioning. Let me just focus on these areas, that how they are actually functioning. So, antibody synthesis, then intercellular pathogenic apoptosis, and then various allergic reaction or mucus production or extracellular pathogen or tissue inflammation. So, these are various function. Now, you may be thinking that sir, you have told that they are actually working like a messenger. Now, how they are doing this kinds of function? See, when it will send its message to the other cell, so during that course some such kind of activities will be happen. Now, tell you more interference. So, a virus infected cell release the interferon causing nearby cells to heighten their antiviral defenses. So, for example, this is a virus, it has infected your this cell. So, what will happen? It will release these things, this rat thing interferon, so that your other cells get ready and this will die, this cell will die, you can see death, it is still dying. So, it will inform to the other cells that you may get alert because some foreign substance actually entered. So, interferons are protein made, they are released by the host cell in response to the presence of the pathogens, virus, bacteria and other things, and they are belong to the large proteins such as cytokine, already you are aware, and the molecules used for the communication, and they also allow communication between the trigger the productive defenses to the immune system to eradicate the pathogen and tumor also. And gamma interferons are produced by the natural killer cell that activate the macrophage like type 2 that is interferon and natural killer cell, and whereas alpha and beta are actually produced by the virus infected cell that activate the natural killer cell. And if I tell you more, here you can see one picture that the role of interferon. So, this is the infected cell, one virus actually invaded. So, it is releasing some interferon and it is actually this interferon informing to this other cell, so that they can get activated. So, this is the that is a proper differences or how the alpha, beta and gamma interference actually happening. I will not discuss this too much because you see this is not a part of your syllabus. I just added these differences for your future understanding. In future, if you do your masters and PhD, this graph will help you. For today's lecture, I will just tell you that their designation and how, where they are actually inducing and their chromosome location. So, formal designation: leukocytes, fibroblast and this is called immune interferon. So, it is actually inducing virus, double stranded virus RNA, and the gamma actually working on the mutations, and their location you can see 22 and 6 chromosome. And chemokines, the third one, chemokines are also one kinds of cytokine. They are a family of the small and secreted structurally related cytokines with a crucial role in the inflammation and immunity. So, when a virus will attack your body, so homeostasis will be happen, then inflammation, and ultimately inflammation and homeostasis will happen. So, these various types of chemokines like chemokines receptor A, B, C and D, they will be come to the picture actually for the just messaging, for the messaging or for the send their information they are working. So, they are also act as a mediator. I will not discuss too much. So, this is a picture you can see how neutrophil are actually releasing and bacteria are actually getting information. This is the last summary of my today's discussion. So, if you can see one microzyte actually coming to the your body, dendritic cell actually presenting, dendritic cell actually presenting, that is endocyte present cell, and you see that is a T cells got the information, they are actually releasing, they are actually proliferating the T helper cells, which releasing the cytokines. Through that B cells get activated, B cells become plasma cells or memory cells, and they are releasing the antibody. Another side CD8, that is killer T cells, and it is actually making the cytotoxic T cells, and traveling through the infection, again cytotoxins are that cytokines are actually forming, and it is creating some inflammation and phagocytosis. So, I hope this, after watching this summary, everything actually so much clear to you. So, that is it from my side for today. Today we have learned that how antigen presenting cells are working, major histochromatic complex, structure, working, and their associated genes, similarity and dissimilarities and cytokines. So, in tomorrow's lecture, in next lecture, I will tell you that hypersensitivity and how our body is reacting with various kinds of hypersensitivity reaction. I will also tell you the various bacterial vaccines including toxics. So, that is it from my side for today. So, I am deeply thankful to you all for joining today's lecture. God bless you all. See you in my next lecture.